Hello students, welcome to Short Observe classes. Today the question is about SDS page. So today the question is about the SDS page. So just look into the question, and the question we have asked here, the molecular weight of a protein. So a molecular weight of a protein as determined by native page is 400 kilodalton. This protein, when run on a ion reducing SDS page, gave a band of 200 kilodalton, and on reducing SDS page gave a band of 100 kilodalton. The protein has which of the following? And see what are the options. That is 4 subunit of which 2 sets are linked by 2 disulfide bonds. Second is 4 subunit which are linked by 4 disulfide bonds or bridges. And third is 2 subunits only and none are linked by disulfide bridges. So none are linked by disulfide bridges. And last one is 2 subunit which are linked by disulfide bridges. Now let me do one thing. I just inserting a page so that I can explain. So native page. Let's know about native page. Native page is everything in tract. Everything in tract. So separated by size and charge of shape. Shape, charge and size is the way through separation. So they separated through this size of charge and the shape charge, the molecular size and the shape charge. Okay, so in the next point, what is SDS page non-reducing? SDS page. These are the little much information that we should know to solve this question. Page non-reducing. Okay. This is sulfide bonding or the bridges that are interact but proteins is denatured. Okay. And now what is the SDS page reducing? That was non-reducing. Now SDS page reducing. Okay. So sulfide bridges that become reduced protein, but still denatured. Protein but denatured. So in a native gel, you don't denature the protein at all. So the 3D shape in the native page become an important determinant to how the protein migrates. This protein's migration is only dependent on the 3D shape in native page. So in a non-reducing SDS page, this was suppose a native page. this was about the native page. Okay. Now, in this SDS page that is non-reducing, what happens? You still denature the protein. You just leave the disulfide bond intact. So, denatured the protein, but this is intact. So the protein will mostly denatured, and if it has disulfides, then those will convey some 3D structure, but very minimal compared to the native gels, not like this much. So if a protein has multiple subunits that are held together by disulfide bond, like this, then those subunits will come apart under reducing SDS. Reducing SDS. Now, in other words, you will see low weight units under reducing condition. The low weight units that are under reducing condition, what happens through this? They are the heavier fragment. They will have the heavier fragment under non-reducing SDS. So this will non-reducing SDS. Now, and if we just run the same protein, suppose the protein is this and the protein is this, and this is the native gel, and this is non-reducing SDS, and if we just run the same protein, so then that can be a heavier. So in this case, in the native gel, this can be a heavier protein or the heavier fragments of protein, or we can get a lighter protein fragment than this non-reducing SDS. So there the thing will, in this question, the answer will be 4 subunits, of which 2 sets are linked by two-sided disulfide bridges. That will be the correct answer for this question. Thank you for watching this video.